All right, why don't we go ahead and get started. So today is our last formal lecture. And recall that on Friday, Dr. Fruman will be back, and he and I will have a review session. And if you guys have any questions about how to prepare for the exam that'll take place on Monday, we can cover that on Friday. So today we're going to be talking about transplantation and this issue of alloimmunity, the fact that the immune system, we've spent a lot of time talking about immune tolerance and how the immune system has no, it's especially T cells are primed to be able to recognize MHC, but there's no tolerance against somebody else's MHC. So we'll go over how that impacts organ transplantation and so forth. And the first thing we'll talk about is the blood group antigens, basically how this can then give rise to a rejection if you don't match the appropriate blood group. So when you're donating blood, you find out what your blood type is, and that then can be used for donating blood to somebody else. And if they don't match the antigens properly, then that can cause problems in the recipient. We'll also talk about HLA and hyperacute rejection. I'll talk about a model, an experimental model for looking at allograft rejection involving mice and skin grafts. And then we'll talk about the different types of allo rejection. So the first type being what we call an acute rejection response that's primarily due to pre-existing antibodies. And then we'll talk about direct and indirect allo recognition, which can give rise to both acute and chronic allograft rejection. We'll talk about minor histocompatibility antigens. And then we'll talk towards the end about immunosuppressive drugs and how they're used to treat graft recipients so they don't reject their grafts. And lastly, we'll talk about maternal fetal tolerance. And at the very end, I just have some comments that I want to make just in terms of scheduling and so forth. All right, so let's talk about transplantation of tissues and organs. And obviously, one of the advantages of this is that, if somebody comes in with some sort of organ failure, whether it's a liver or a kidney, in the past, before transplantation was available, those people would often not survive for very long because that's a major organ system. You need to have that function. And by being able to transplant that from an otherwise healthy individual, then you can then replace that lost function. And one of the things I want to point out is that now we know about stem cells as well. So I'm sure you guys have all heard about stem cells and some of the problems and promises with that. One of the things that we have to think about with respect to stem cells as well is that while they may be able to promote new cell growth and try to promote repair, we also have to worry about transplantation issues there. So if that stem cell is coming from a different individual, then there's a concern about it also causing a rejection response. And this is actually something that my lab is working on quite a bit these days, trying to figure out how stem cells can be used to revitalize the brain after demyelination. So obviously one of the problems that is encountered is that because of the fact that MHC from somebody else is not present in our thymus when our thymocytes are going through the process of selection, there's really no information there to say whether that's something that should be recognized, whether it's considered a self-antigen and so forth. And in a sense, these allografts are considered foreign antigens. They have lots and lots of foreign antigens. And so when we talk about an alloreaction, what we're basically talking about is one T cell basically being able to recognize another T cell simply based on the disparate MHC. So if there's a different MHC, whether it's class 1 or class 2, that, because of the fact that it's not been tolerized, T cells in a recipient can recognize that and then attack it. And so we'll talk about some examples of that, how that is. And the reason for that recognition, number one, is because obviously MHC is polymorphic, as you guys have learned. So everybody, look at your neighbor. Your neighbor, let's say that they decided that they were going to be nice and give you a nice Christmas present because your kidney was not functioning properly. And, well, it's probably not likely, but in any event, let's say that they wanted to donate something to you because you've been such a great colleague during, during this course. So obviously, the one thing that you'd want to first make sure of is that their MHC is really, really close to yours. And so if the person next to you happens to be your identical twin, then no problem, right? But obviously, most of us are going to have different MHC alleles from the person sitting next to us. So the issue there is that our T cells, because they're really good at recognizing MHC, and number two, there's no selection against recognition of your neighbor's MHC, you have T cells that are going to be able to recognize that MHC and then basically think of that as a foreign antigen, okay? And, as I mentioned before, the T cell receptor genes have been, they've gone through evolution, they've been designed to be able to bind very efficiently to MHC. So, if they bind very avidly, then the result is that you can have a rejection response. Okay, so that's kind of the issue that we're going to deal with today. And one of the things that's involved, and one thing I want to point out is that of all of our T cells, it's estimated somewhere between 1 to 5% are overtly alloreactive against a particular MHC. So that's way higher than the the number of cells that might be specific for, say, an antigen or something like that. So our immune system, our T cells, are really designed to be able to bind MHC. And there's a lot of them that could, be activated and then cause a transplant rejection. So one of the issues here, obviously, is trying to match MHC, and we'll talk about some techniques to do that. But another problem, obviously, is just being able to find a tissue transplant. And an example of this is provided in your text, where basically a traffic accident here provides a kidney for transplantation. And then the kidney is then flown to a transplant center. So typically people that need a transplant, they're going to be typed. They're going to look for what their MHC is, and then they're going to try to look for a match. And so they're going to, there's a whole national database where they have information for this for somebody that needs a kidney transplant or what have you. And then once they find an appropriate match, and unfortunately, for kidney transplant, the person doesn't have to die in a traffic accident, but they have to be willing to donate one of their 2 kidneys. In any event, the person is called in the middle of the night, then they then will conduct the transplant surgery. And actually, even before this is taking place, they have to do a lot of things to make sure that that person is not going to overtly reject that transplant. Now, it's sort of a kind of gross, I don't know if it's humor, but it's sort of a shocking. I don't know if you guys have ever heard of Freakonomics before or not, but there's a really interesting book, actually several books now that have been published, talking about all sorts of weird things, in particular using an economic bent on things. And in this, they talk about the number of tissue transplants that are available. So, as a result of the motorcycle helmet laws that have come into play in the 90s and last decade, the number of transplants has actually gone down quite a bit. And I'm not trying to make any particular thing about motorcycle riders. Anybody here a motorcycle rider by the? So nobody, okay, well, all right. In any event, when those helmet laws have been in place, it's reduced the number of transplants because a lot of times the transplant needs to be healthy, so it needs to come from a healthy individual who just happened to die in an accident, which is obviously unfortunate. One interesting note that they point out with this Freakonomics is that when they repeal in certain states, the number of transplants considerably increases. And you can see the little motorcycle flying off the end of this graph here that sort of, points that out. So, as they say to parents, give your son a motorcycle for his last birthday. Anyways, all right, enough. So the point is that, finding transplants is difficult, they're rare, and so if you're going to do a transplant, you want to make sure that you're going to do the right job and that transplant is going to survive. All right, so the first type of transplant that I think we're all familiar with is the blood group antigen. So if somebody, say as a result of an accident or something, they've lost a lot of blood, they need a blood transfusion. In order for that to be successful, the physicians need to make sure that the blood group antigens are matched. And so an example of this are the O, A, B, and AB types. And basically that's all really based on the kinds of molecules that are expressed on the surface of red blood cells. So on the surface of these, there are proteins and lipids that have differential glycosylation. So they have these sugar molecules on the surface of some of these receptors. And basically, all of us have what are called natural antibodies. These are primarily of the IgM type that our B cells will make. And if you look here, and somebody has group A antigen, so they have this group A antigen on the surface of this red blood cell. in that particular individual, you don't have any anti-A antibodies, but you do have anti-B because there's no tolerance here, right? You don't have the B antigens, and as a result, you don't have the B antigens, and as a result, there's no tolerance against B, and so you have those natural antibodies. There's no selection against that. Conversely, in somebody that has the group B antigens on the surface, you don't have type anti-B antibodies, but you do have the anti-A antibodies because, again, there's no tolerance against this. And if you look in folks that have A and B, this group AB, they have neither the type A or type B antibodies. And then for group O that have essentially no glycosylation on these receptors that can produce these natural antibodies, they have both A and B. So in my case, I'm group O, so I'm what's called a universal donor, so I can give my blood to anybody, but I can only accept group O blood because I have antibodies against A and B. All right, so that's one important thing that you need to make sure of. Now, a second kind of, this is a model that has been basically used to look at the whole process of allograft rejection. And it's based on doing skin transplants in mice. And I should say that this is a very stringent assay, so you can get very strong allorejection. And it's based on the idea that if you take mice, and in this case it's a yellow mouse here, which has one type of MHC. And I should say that all these mouse experiments that we've been talking about are typically done with what are called congenic mice. These are mice that have been they've been back-crossed many, many times, so they're essentially genetically identical. So in a litter of mice, all of them have exactly the same genes. So if you take a mouse that has this MHCA and you transplant that onto another mouse that's of that same genotype, that graft then is tolerated because the MHC is identical. So you don't have any differences in MHC. And so there's no allorejection. If, on the other hand, you take a mouse that has MHCA and you take its little patch of its skin and transplant that onto this blue mouse that has MHCB, you get a little UCI mouse here. And then on top of that, the little graft is going to get rejected very quickly because that graft is fully mismatched, both in MHC class 1 and class 2. And so within a little over a week, you start to get full rejection of that allograft. Now, if you take that, basically that same mouse that had been the recipient of that skin graft, and then you transplant some fresh skin from this MHCA mouse, you get an accelerated graft rejection. And the reason for that is that you have memory cells there, right? So you have T cells that, as a result of having previously undergone clonal expansion, those memory cells then can become activated, and you get this accelerated rejection of the graft. So here, instead of having the graft rejection about 10 days, here it's maybe about half that time. So it's an accelerated rejection because of those memory cells. And we know that the T cells are important in this because if you take one of these mice that's been sensitized and it's rejected one of these allografts, the skin allograft, and then you transplant that into a naive mouse that's never seen this allogeneic skin graft in the past, and then you transplant a skin graft onto that mouse, you also have that accelerated graft rejection. So that basically shows you through this sort of adoptive transfer experiment that the T cells are really important in mediating this kind of skin graft rejection. Okay, any questions about this skin graft allorejection? Okay. All right, so basically when we think about allograft rejection, we basically think of two different processes, one of which is transplant rejection, which is basically what I've shown you with respect to skin transplant rejection. And then there's another thing that's called graft versus host reaction, or graft versus host disease. And basically the first example, as we've talked about, if you take somebody else's kidney in this case, you transplant it. If there's not a good match between the MHC and the host kidney and the recipient's body, then you will get a rejection response against that kidney. And another example is this graft versus host response. So in this case, let's say this is a bone marrow transplant. So let's say that somebody has leukemia or something like that, and then they get a lot of chemotherapy that kills off their all their lymphocytes and other cells, other white blood cells. oftentimes those folks will get a bone marrow transplant from somebody else. And so again, you want to make sure that there's a good match between the bone marrow donor and the recipient of that transplant. Otherwise, you'll get a rejection response. But on top of that, the bone marrow actually has quite a few memory T cells. And so in this case, in addition to being concerned about the recipient rejecting this bone marrow transplant, if there are T cells that are coming across in the bone marrow transplant, they can actually start to attack the host tissue, because now all of a sudden they're surrounded by this massive amount of foreign MHC. And so that basically, the allograft in this case is actually recognizing the host. All right. So let's think of a couple of terms to think of here. One of which is called an autograft. And so this is basically, as it implies, if you take a transplant from one side of the body and then transplant that elsewhere, that will then, obviously because the MHC is the same, you're not going to get one of these sort of rejection responses. And an example of that is a skin graft. So, sometimes if people get a burn or something like that, they may have to take a piece of skin from a different part of the body and then engraft that to try to deal with, that barrier function. And then another term is what we call syngeneic or an isograft. And this is basically where the recipient and donor are genetically identical. Certainly that is true for the MHC loci. And that would be a situation where, let's say you have a transplant between somebody that's genetically identical. It could be an identical twin or similar, just a regular old twin. But the point of that is that they're going to have the same MHC, and if they're MHC matched, you're not going to get a rejection response. And then everybody else, which would be give rise to a situation where it's allograft or it's allogeneic. So allogeneic simply implies that there are differences in all of the haplotypes of MHC. Now, one of the ways that you can measure this, there's a couple of ways. So if you're a transplant physician and you want to do a transplant into somebody that needs, let's say, a kidney, what you'll do is you'll do a whole panel of antibody matches to look at what MHC haplotype that individual has. And so that takes advantage of something called an ELISA assay. But another thing that you can do that's fairly useful is something called a mixed lymphocyte reaction, or an MLR. And that's based on the idea that if you take lymphocytes from one individual and you mix them with stimulator cells, these could be various antigen presenting cell types from another individual. If the two individuals are syngeneic, then you're not going to get a response. But if they're allogeneic, the T cells are going to start to respond and become activated. So basically the way this works is you have these, the T cells, which in this case we call the responder population, because they're going to respond to this disparate MHC. And then you have the stimulator population, which are the antigen presenting cells. And then you can mix the two. And oftentimes when we do this with mice, for example, we just take like whole spleen, munch those up and get a single cell suspension, and then mix those two populations. But one of the things that we have to do to make sure that we're going to get a good mixed lymphocyte reaction is to make sure to either irradiate or in some way inactivate the stimulator population. Because if the stimulators are capable of proliferating, there's going to be some T cells there. And those T cells are going to also be able to recognize some of the MHC that's present on the responder population. So you have to basically set up this one-way sort of mixed lymphocyte reaction, because you want to see if this population is going to respond or not. If you don't do that, then the T cells from both sides are going to try to kill each other, and you're not going to get much of a MLR response. Okay, and there's two ways that you can measure the outcome of this. One is to measure T cell proliferation, so you can look at whether those T cells are responding. Oftentimes that's done using uptake of tritiated thymidine, for example. And then a second way is you can measure cytotoxicity. You can actually look to see whether these responders are killing some of these stimulator cells or not. All right. So any questions about mixed lymphocyte reaction? All right. Okay, now there are different types of rejection responses that could potentially take place. And the first one, as a transplant physician that you're going to be concerned about is what's called the hyperacute rejection. And this is basically going to occur as a result of having pre-existing antibodies. And a good example of those kind of antibodies are what we just talked about, these blood group antigens. So the first thing you want to make sure is that the donor and recipient have the same blood group antigen profiles. Because if you don't do that, then you're going to have some of these natural antibodies that can then respond against the graft and then cause problems. And so because of the fact that these are pre-existing antibodies, you don't need any sort of clonal expansion or anything to take place. This then results in very rapid rejection within 24 to 48 hours and involves complement fixation and the destruction of the tissue. So that's shown here where you have a kidney. And this kidney, let's say that this is from one individual that has type O blood, and then this is going into somebody else, or let's say type A, and this is going into somebody else that has type B blood group antigens. And the result of that is that this, the recipient is going to have pre-existing antibodies against the disparate blood group from the donor. And so the consequence of that is that there are going to be these antibodies. They're going to start to then attack the various cells in that allograft, including the vasculature. And so in this case here, you can see that there's antibodies that are binding to some of these endothelial cells in a blood vessel inside of the allograft in the kidney here. That's going to then result in complement fixation. And then, just like we've talked about with respect to some of the other or the hypersensitivity responses, that's going to then induce this sort of inflammation. And ultimately, that's going to lead to occlusion of that blood vessel, and then that allograft is going to die off and fail. So that is the first response. So the hyperacute response is something that can happen within the first few hours of transplant. The second is what's called an acute rejection response, and this is something that's likely to happen over the first, essentially the first week or so. And it takes a bit of time for this to develop because it requires T-cell clonal expansion. And so, and this is an example here of a kidney. It kind of looks like haggis. I don't know if you guys have ever heard of that before, but anyways, sorry, no more bad jokes. This is the last lecture, so I promise no more. All right, so no, I can't do that. Anyway, so this is an example of a kidney that's undergone this sort of acute rejection response, and this is happening over several days. Basically, T cells here are recognizing the antigens in this allograft as foreign, and then you start to get a T cell response against that. And so ultimately then that graft is going to fail. And this can be blocked, as we'll talk about, using what are called immunosuppressive drugs, certain drugs that can block inflammation and or T cell responses. So how does this acute rejection take place? So there's two different types of allorejection involving T cells that we'll talk about, what's called direct and indirect. And so the first is direct, which makes, which is easy to think about. Basically, here you have a transplant, kidney transplant, and it has associated with it some of these graft-associated dendritic cells. And so some of the dendritic cells that are inside of that kidney, once they hook it up to the host, to the recipient, some of those dendritic cells can then make their way through the blood vessels, find their way to a nearby lymph node, and then the consequence of that is that you activate those alloreactive T cells. They can then travel back into the kidney and then start to attack that graft tissue. And so the consequence of that is that over time, that graft then starts to die off. The T cells start to attack the endothelial cells and so forth, and then ultimately the graft fails. Okay, so that's called direct allorecognition. And the point of this is that the T cells in this case are being activated by these dendritic cells that have this foreign MHC. So they're directly binding to that foreign MHC and recognizing that as being foreign. Now that's just different from something called indirect allorecognition, and this is something that takes place typically in as a response to chronic allorejection, as we'll talk about it in a minute. But the point of this is that this indirect allorecognition doesn't require these dendritic cells or other kinds of antigen-presenting cells to be present in the allograft. Even if you completely eliminated all of these antigen-presenting cells from the allograft, you would still have to be concerned about this indirect allorecognition, which is basically where you have, let's say, some dying cells that are present in the allograft, which you're always going to have some of that as a result of the stress of surgery. Those cells are then going to shed off some of their MHC, whether it's class 1 or class 2. It's going to then get engulfed into the recipient's dendritic cells. The MHC will then become processed into antigenic peptides and then presented on the recipient's MHC on the surface of those dendritic cells. And so in this case, these allospecific T cells are not directly recognizing an intact piece of MHC on the surface of a cell, but instead they're recognizing peptides that are processed from the donor's MHC. And so the consequence is the same. You activate T cells, and those T cells can then mediate a T cell response. And so in this case, here we're showing a CD4 T cell recognizing some of this foreign MHC peptides. And the consequence of this is that T cell can then become activated and lead to chronic rejection. Okay, so does anybody have any questions about direct versus indirect allorecognition?
Yeah, so indirect, does it have to be from an APC? Could it just be from any cell?
Can indirect be any particular cell? Does it have to be an antigen presenting cell? Yeah, it can be any cell. So obviously there's a whole bunch of class 1, and if you have processing of this allogeneic MHC class 1, you're still going to get presentation. You get activation of T cells, either CD4 or CD8, because indirect, they're recognizing peptides. They're no longer recognizing the structure.
Whereas direct has to be from an APC.
Correct. Any other questions? To prime a response, that's true. Any other questions about direct versus indirect? Okay. So chronic rejection, so you have this hyperacute, which takes place within the first few hours, this acute response, which is taking place within the first few days out to the first couple of weeks or so, whereas chronic rejection is something that can happen over the order of years. So, and this is particularly the case where, perhaps through indirect recognition, you get a bunch of T cells that can make their way inside of the allograft and are activated against the allograft MHC, and then start to attack some of these endothelial cells that line the allograft. And the reason why oftentimes those are the first to get attacked is because all of these different tissues that are being transplanted, oftentimes they'll have lots of blood vessels in order to be able to to supply oxygen to the cells in that particular organ. And so over time, you start to get this thickening of these blood vessels because of this inflammation. I'll show you an example of that in the next slide. But the result of that is that you start to lose blood flow into the tissue, and that's called that's one thing that we call ischemia. So basically, because of the fact that you have this constant inflammation going on, over time, you're not getting blood supply, and that starts to starve the tissues, and ultimately the graft will fail. And one important component for this chronic rejection is the antibody response, and that's particularly the case against these class 1 MHC antigens that are then going to be processed and presented to these T cells, and the T cells can then act as helper cells to be able to promote the generation of stronger and stronger class 1 alloantigen specific antibodies. And so that's shown here, where you have, let's say here's a T cell interacting with the dendritic cell inside of the recipient. The recipient has taken up some of the graft MHC because some of the cells have died off or what have you. Those then make their way, they're processed into peptides, and the peptides make their way into the MHC of the recipient dendritic cell, which then can stimulate this CD4 T cell. And then that CD4 T cell can interact with the B cell, which is making antibodies against the donor's class 1, which is going to be presented on the surface of cells, including the endothelial cells on these blood vessels that line the tissue. And so the consequence of that is that those alloreactive antibodies are then going to cause this inflammation to take place inside of that tissue, and then you're going to get ultimately graft rejection. And so that's shown here, where you can see what's happening over time. You have these alloantigen specific antibodies, and they're recognizing, for example, class 1, which is going to be presented on the surface of pretty much all the cells inside of the organ, but in particular have really good access to these endothelial cells in these blood vessels. And so the consequence of that binding of these MHC specific antibodies is that just like we talked about before when we were talking about some of the hypersensitivity responses, the result of that is that you're going to start to activate some of these inflammatory cells, like mast cells and granulocytes. You're going to have T cells that come in. And over time, and this is the reason why we call it chronic, and it takes quite a bit of time, is because these reactions need to take place over quite a bit of time. And the result of that is that you start to get this highly inflammatory situation. Those inflammatory mediators can then cause thickening of these blood vessel walls, and ultimately this then occludes the blood vessel. You lose the blood flow, and then of course, if the organ is ischemic, it doesn't have enough oxygen, it's going to die off, and so that then will lead to this sort of chronic rejection. Okay, any questions about tissue damage induced during chronic alloimmunity? All right. So one other thing that we talked about a little bit before are what are called minor histocompatibility antigens. And the point of this is that, even if you had a perfect match between the donor and the recipient, you still have to worry about these minor histocompatibility antigens. These are, and this makes it a lot harder to get a perfect match between one donor and a recipient. And the reason for this, what are these things? So basically, these are essentially any particular gene that could be expressed that is polymorphic. So you guys have learned about polymorphism in the context of MHC, but there's a lot of other genes in our bodies that have this kind of polymorphism as well. And so if there's any of this sort of polymorphism that's taking place, and those are different between the donor and the recipient, then you could have, you could activate some T cells. And examples of that, let's say, here you have a donor, and it's processing all the time self-peptides, which then make their way on the surface of these donor cells. But of course, in the recipient, they're going to have, their own set of proteins that can be presented by MHC. And so if you get some of this, some of these polymorphic proteins that are being expressed on the donor cells, that can then be presented to a T cell, and you will get a response. And one example that we talked about is something called this male HY antigen. So the male HY antigen, and I mentioned before when I was talking about this particular T cell receptor transgenic mouse that recognizes that during thymopoiesis. And so in the males, you had this really strong negative selection, and in the females, you had strong positive selection. So that is an example of a minor histocompatibility antigen. And one point that I want to make about these guys is that the response against them is nowhere near as profound as you would have with direct allorecognition. The reason for that is because these peptides are, they're peptides. They're not MHC. So the T cell receptor is really good at binding to MHC, but for the recognition to take place against these peptides, it's not, you don't have as many clones that are going to be capable of recognizing that. So in other words, it's going to be this is going to give rise to more of a kind of a chronic allorejection response than you would have against, say, a direct recognition where you have direct recognition of intact MHC molecules. Okay, any question about minor histocompatibility? Okay, so now you've identified a transplant recipient, you've identified a transplant donor, and oftentimes you're not going to be able to match everything. You're not going to be able to match either the MHC. You try to do your best and try to minimize the number of haplotype differences, but you're never going to be perfect unless they're from identical twins. So, and then of course you have to deal with all these minor histocompatibility antigens as well. So what do you do? I mean, obviously you could say, well, we're not going to do the transplant unless we get it perfect, but obviously that's very rare. So instead you have to turn to immune suppression. And so immune suppression is basically this approach that you want to take to block T cell responses and to block B cell responses such that you don't get these kind of rejection, this allorejection taking place. And one example of immunosuppressive drugs are what are called corticosteroids. And basically what these guys do, so first of all, the reason why they're used but they're not particularly popular is that they have a lot of off-target effects, and they can be toxic to various tissues and organs, which oftentimes will sort of limit the duration of a transplant. Oftentimes because you're suppressing the immune system, and then there are these off-target effects, these transplants may not, or the transplant recipient may not survive for as long as they might want to. So the corticosteroids, they have a variety of different ways that they work. One example of this is that they target the NF-kappa B pathway. So you guys learned about NF-kappa B as a transcription factor, both in the context of innate immune signaling, how toll-like receptors, when they recognize a PAMP, they activate NF-kappa B. And we also talked about that in the context of T cell activation. So kappa B is an important transcription factor that promotes T cell activation. And the way that it does that, I'm not going to go back through the pathway, but basically NF-kappa B is the transcription factor, and it's bound to something called I-kappa B. So I-kappa B is the inhibitor of kappa B, and basically it binds to the NF-kappa B transcription factor and keeps it from going into the nucleus. And so obviously a transcription factor that's not in the nucleus is not going to have, it's not going to be impacting gene transcription. So these steroids can work by upregulating the expression of I-kappa B and basically blocking NF-kappa B activation. So how do they work themselves? So here's a steroid. So these are basically little fatty lipid molecules that can make their way through the body, through the blood to a cell. They can then make their way across, because they're kind of fatty, they can make their way across the membrane of the cell into the cytosol. And there they interact with specialized receptors called steroid receptors. And again, in the absence of a ligand, these receptors for steroids are bound to something called heat shock protein 90 or an HSP. Once they bind to this ligand, they then displace this heat shock protein, and at this point, they can then go into the nucleus. And so again, just like with NF-kappa B, once they're in the nucleus, they can interact with promoters in front of genes and induce gene transcription. And so, in this case, the gene in this case would be this I-kappa B. And so by binding to this I-kappa B promoter, these glucocorticoid receptors can then induce I-kappa B expression and basically shut down immune responses, both in innate immune cells as well as in lymphocytes. Okay, so that's one example of an immunosuppressive drug. And these corticosteroids are quite effective because they have so many targets, both innate and adaptive. And so examples of this is that they can reduce the expression of pro-inflammatory cytokines like interleukin-1 and TNF, GM-CSF, which can promote monocyte differentiation in the bone marrow, all of these different cytokines and chemokines, and can then reduce inflammation. This also can lead to the reduction of something called NOS, or nitric oxide synthase, which is involved in the production of NO, nitric oxide, not the stuff you get when you go to the dentist. So this stuff is basically, when we talk about macrophages and various other innate immune cells, this is very toxic to bacteria. So if you reduce the expression of the synthase, you reduce the expression of NO, and so in addition to being able to kill bacteria, it's also a pro-inflammatory mediator. So you're getting rid of that, so that helps to reduce inflammation. Also reduces the production of these molecules that are involved in production of prostaglandins and leukotrienes that again are pro-inflammatory. Reduces the expression of adhesion molecules, so you don't get migration of leukocytes into the tissues. And then also importantly, induces the expression of what are called endonucleases. And these guys can then lead to the apoptosis of lymphocytes, T cells and B cells, and to granulocytes like eosinophils. So the consequence of all that is that you reduce inflammation, by blocking all of these pathways, and you also promote the apoptosis of leukocytes and lymphocytes, the consequence of which is that you can then control the response. But again, the downside to these corticosteroids is that in addition to targeting the immune system, they also target a lot of other cells because these steroid receptors are present all over the body. All right, so that's one downside to corticosteroids. And of course, they're still used quite a bit in the clinic, but we obviously want to have better drugs. Okay, so any questions about the corticosteroids? All right, so another example of a immune suppressive drug or a target of immune suppressive drugs is something called NFAT, which is another transcription factor that we've talked about. And this particular transcription factor is activated for example, in T cells. So here you have a T cell, here's its T cell receptor interacting with MHC peptide, the consequence of which is that you get activation of calcium mobilization inside that cell. So calcium basically starts to become elevated in the cytosol. The result of that is that then activates NFAT. And I'm not going to go through all the details of how that works, but basically when there's calcium present, NFAT can become, it can then make its way into the nucleus and induce gene transcription. So obviously this is an important way that transcription factors can gain activity. Do we have a question?
On the slide before, the corticosteroid receptor, so is there an analog like this for that receptor in us, like innately? So is there a way that we can suppress the immune system on our own without having corticosteroids being injected in us? Is that why we have a receptor?
Oh yeah, I mean the corticosteroid receptors, that we have a lot of endogenous corticosteroids and other types of steroids that are produced by the body for various reasons. Whether those are involved in some sort of innate regulation of lymphocyte activation or inflammation is a good question. But there are a lot of targets for those, and oftentimes the steroids are produced as hormones that can have kind of long range effects. Any other questions? All right. So NFAT is activated in response to calcium mobilization, and basically there's two different classes of drugs, and they kind of work the same way to block NFAT translocation into the nucleus, which is shown here. And one of these is called cyclosporin A, or CSA, and basically it interacts with inside of the cytosol with a protein called cyclophilin. And so when it interacts with cyclophilin, the result is it basically blocks the ability of NFAT to make its way into the nucleus. And another class is called FK506, which instead of interacting with cyclosporin or with cyclophilins, it interacts with what are called these FK binding proteins, or FKBP. But both of these, cyclophilin and FKBP, are important in this target. And when they're bound to their drugs, they can block this translocation of NFAT. So by blocking NFAT from getting into the nucleus, NFAT is a key transcription factor in T cell activation. So the consequence of that is that those T cells can then not make their way into the the whole process of clonal activation, and so this is a reasonable way to block the immune response. So I wanted to quickly show a little movie on another way that people are now targeting adaptive immunity.
Success rates in solid organ transplantation continue to rise. Yet each measure of success brings with it a new set of challenges. Signaling the future, targeting co-stimulatory pathways. The quest to improve long-term outcomes begins with an exploration of the initial cascade of events that occurs within the immune system. Once an organ is transplanted, the body mounts an immune response against the allograft. T cells, which mediate the immune response, require two signals for full activation. Signal 1 is mediated through the T cell receptor's engagement with the antigen presented by the MHC on the APC. Signal 2, the co-stimulatory signal, results from receptor-ligand interactions that occur between the T cells and the APC. This co-stimulation enables T cells to proliferate and mount a sustained response to the allograft. Although current immunosuppressive therapies, such as calcineurin inhibitors and mTOR inhibitors, work to prevent T cell activation and/or proliferation, their targets are not specific to the immune system and may be associated with therapy-related toxicities that may compromise long-term graft and patient outcomes. To meet the current challenge of reducing toxicities and prolonging graft and patient survival, selective agents are needed. While multiple pathways are currently being investigated for the development of immunoselective agents, CD28 is one of the best characterized receptors required for co-stimulation. By preventing CD28 from interacting with CD8086 on the APC, signal 2 is blocked, and the T cell is rendered anergic or apoptotic. Since CD28 is expressed predominantly on T cells, this pathway offers the potential for immunoselectivity, possibly reducing the toxicities that may be associated with less immunoselective agents. Co-stimulation blockade via inhibition of the CD28 pathway is a therapeutic approach with the potential to improve long-term graft and patient outcomes, and one that warrants further investigation.
All right, so but the point of that is basically that when you have these kind of inhibitors like corticosteroids or these, FK506 and cyclosporin, the disadvantage is that, as they mentioned in the video, is that there's a lot of different cells that have, these receptors for corticosteroids. There's a lot of different cells that use NFAT for signaling, and so, the problem there is that you're going to have a lot of off-target effects. You're going to have impacts in other tissues. So the advantage of co-stimulatory blockade is that that's specifically targeting naive T cell activation. So that's a great thing. But even with that, there's a problem, which is that, any kind of antiviral response that you might want is going to be impacted by that. So there's still a lot of work to be done to try to treat this problem. And just the last point that I wanted to make with respect to these allografts is that there's different survival rates for allograft transplants with these kind of immune suppressive drugs. And some of the best examples in terms of 5-year survival rates are with things like kidney. That sort of drops with liver, and is particularly a problem with the lung. And it probably has to do, at least partly, with the fact that the lung is just got so many blood vessels associated with it that are involved in exchange of oxygen. So there's still a lot of work to be done in this area. So the last point that I want to make has to do with maternal fetal tolerance, right? So if we think about what is the allograft that's most likely to be there that doesn't require any sort of transplant or anything like that, and that is this little fetus. So you get this blue guy and this pink woman together, and they make this little fetus. And of course, that fetus has not just the mother's MHC antigens, but also the father's MHC antigens. So there's got to be something going on that allows this mother's immune system to tolerate this allograft, essentially, right? These folks don't have to get, MHC match before they get married by the pastor or whatever. So the way that the body deals with that is that the placenta happens to be what we call an immune privilege site. So it sets up essentially an immune tolerant situation to protect the fetus from T cells that are maternally derived from attacking the fetus. And so some examples of this, the placenta itself is devoid of class 1 and class 2 expression, but at the same time is not targeted by NK cells. So it has special tolerance against MHC and NK recognition. There's also a bunch of suppressive cytokines like interleukin 4 and 10 that are expressed by the epithelium of the uterine, which then can prevent Th1 responses, as well as the placenta is starved of certain nutrients that are required for T cell activation, like tryptophan, and so the consequence of that is it prevents these responses. And so one of the keys is that the fetus is tolerated through two processes, one of which is that it essentially occupies what we consider a non-immunogenic site, and so it has a sort of immune privilege. And on top of that, there's a local immunosuppressive environment that prevents any sort of overt immune response. And so there's still a lot that we don't understand about this maternal fetal tolerance, but that's what we do know. All right, so I just want to take a couple of minutes and talk about the next couple of days. So as I mentioned before, on Friday we'll be having a review session. Dr. Fruman will be back, and roughly about 25% of the final exam will be from his part, so you can direct some of the questions to him about that. I've uploaded both a study guide as well as a practice final, and so any questions about the sort of things that you're going to see on the exam, they're really going to be addressed in the study guide as well as that practice final. I'm having office hours today at 2 o'clock, and if you can't make it to that time, again, email me. We can try to set up a time over the next couple of days. Unfortunately, we don't have time next week because it's, the exam is on Monday. So in addition to that, please take advantage of the message board, which is online. I've only gotten, I think, one comment I got today. So feel free if you have questions that you need to be addressed. I'll try to do that, try to do my best to answer them. Don't expect, answers in the middle of the night or, a half an hour before the exam, because I'm probably going to be busy. So it's probably more, so take advantage of that, but within reason. And then I want to point out on this spring, if you're really interested in immunology, we offer a course called M119, which is an advanced immunology class in the spring quarter, and Dr. Fruman and I will also be teaching in that class. And that's more, more advanced, obviously, the name. But it's also, we do some, much more cutting edge stuff, so things that are coming out. And it's a much smaller class, so, we'll have a greater chance to interact, if that's a good thing, I don't know. The last thing I want to point out is that we have online evaluations that have been activated. Those are going to be open for the next few days. And each evaluation that you turn in is worth 1 extra credit point. So one for me and one for Dr. Fruman. And the way that we apply the grades, we figure out the grading before we apply those extra credit grades. So oftentimes, and this, it actually can affect your grade. It can push you up. So if you like the class, remember my name is Dr. Walsh. If you didn't like it, I'm Dr. Fruman. Okay. All right, so we'll see you guys on Friday.